So the next session, my name's Alison Cave, I work within the Pharmacovigilance Department at the EMA, and the next session is going to try and focus on what were some of the success factors for our multinational collaborations among registries. And the session is going to be kicked off by Carmen Martos, who's from the European Network of Cancer Registries from Italy, and she's going to talk about standardization of cancer registries, data collection and validation at a European level. Thank you, Carmen.
First, I would like to thank to the organizer of this very interesting workshop to invite us to present and to share with you what we are doing at the European level with the cancer data. First, I'm going to give you an overview what is the GRC and what is the European Network of Cancer Registry. I'm working at the GRC, the Joint Resource Center, is Directorate General of the European Commission, is the European Commission's science and knowledge service. It employs around 3,000 staff. Most of them are researchers and carry out research in order to provide assign independent advice and support to the European Union policy. It's split in size in 5 countries, and our group, the Cancer Information, is working in ISPRA, Italy. We are in the Health in Society unit, in the Directorate Health Consumers and Reference Materials. In this unit, there is other working group, Nutrition and Health, Rare Diseases, Healthcare Quality, and our group, Cancer Information. And our mission is to provide up-to-date statistics on cancer incidence and mortality across Europe. One of the commitment of the European Commission is to reduce the burden of cancer in Europe, and for that we need accurate and comparable European cancer data on incidence, prevalence, survival, and mortality. Our group, at the moment, we are working mostly of the time to standardize data collection and to have this data comparable across Europe. And also we are focusing very much in the quality of this data. We are working for this commitment. We are working together with the European Network of Cancer Registries. This network was established in 1990, connected European population-based cancer registry. I would like to highlight this population-based because this registry, population-based cancer registry, seeks to collect information on all new cases occurring in a very well-defined population. Usually this definition came from the geographical area. And then that means collecting data of cancer about from this resident in this specific area. The main objectives of this network is to promote collaboration between cancer registry, to define data collection standards, and to support cancer registry staff, and also to disseminate information across Europe on incidence, mortality, and survival at population level. This network is governed by the steering committee, and it is supported and coordinated by the GRC's Cancer Information Group, where I'm working. It is affiliated to the International Association of Cancer Registry. We are supporting the ENCR, the European Network of Cancer Registry, since 2012, and serving as ENCR Secretariat. In this framework, we are coordinating, we are organizing the steering committee meeting, the other activities, for example, workshops, working groups, etc. And we are also established or re-established the ENCR plenary conference, where all the European Cancer Registry, or most of them are participating, and the last one was in October in Baveno. Also, in this collaboration, in this framework, the last year, in June 2015, we launched, GRC and ENCR launched a call for data, and we invited the population-based cancer registry in Europe to send the data to us in order to produce indicator on cancer incidence and mortality. For that, first, we established a data collection or data summation portal in order to get the data in a very secure way. Also, we prepared the data collection protocol in order to have or to get the data in a standard way. Also, we prepare also a questionnaire related to this data collection in order to better interpret the data from the cancer registry. And previously, also, we developed a data cleaning software, that this software should be used for all cancer registry and for the European project. At the moment, we got the information from 107 registries across Europe from 99 countries. That means we have collected already about 24 million of cancer data. In orange, you can see the coverage of the cancer registry that sent data to us, and in green, there are some registries that we are still trying to collect data from them. In gray is areas where they are not covered by the population-based cancer registry. Focusing now in the data, cancer data related to treatment, in this protocol, we define some core variable that is necessary to produce information about incidence in Europe. And then, for example, age, or date of birth, sex, also very detailed information about the tumor, topography, morphology, behavior, grade, follow-up, etc. And we included in this question, in this call for data protocol, some information about treatment as optional variable. And we asked very basic information about treatment. We asked if the patient had, but first we define which mean for us treatment for this data collection. And for us, it was the first course of treatment, anti-cancer treatment after diagnosis. This is the information that we are collecting in this call for data. And then we asked if the patient got surgery, systematic therapy, radiotherapy, and bone marrow transplantation. Only if yes or not in the first course of treatment. From the questionnaire that I mentioned before, we have 3 questions related to this data collection treatment. And we can see that 54% of the cancer registry are collecting information about treatment. But only 10 of these cancer registry are collecting more information, more detailed information that we asked for. And even if some registry are collecting information on treatment, they didn't send to us because they said the information is not good quality or is incomplete. The information sources for collecting this information on treatment mainly is clinical records, notification by clinician, or both clinician records and notification by clinician. In this situation, we can see that usually or routinely, the population-based cancer registry don't collect information, detailed information on treatment. But we think that population-based cancer registry are potentially a valuable source of information to evaluate cancer care quality and treatment effects on the real world and practice. Because we have to remember that the information is a population level. It's not for a selected patient sample. Since there are no standards for treatment data collection and coding, this information is not collected routinely by the cancer registry. The information they are collecting maybe is not comparable one country to another country or among registry. If the information is available in the clinical records, we think that they could be improved the data collection on treatment by the cancer registry. But we need something to have this or to improve this data collection. It's necessary to improve or to provide them the rules or recommendation to harmonize the data collection and coding for treatment. Cancer registry staff also should be supported to identify the different drugs given to the patient, because when the cancer registry staff go to have or to collect this information on the clinical record, usually they found the commercial name in these reports, and then it's difficult for them to identify which drugs have to be collected, how to classify as target therapy, monotherapy, chemotherapy, etc. We prepare a report in order to standardize the data quality check among European Cancer Registry, and the Bulgarian Cancer Registry made a table very useful for their staff to have. They have included the AD code, the commercial name, and also the generic name for this. But this table cannot be used around Europe because could be different. At the moment, just a pilot study in order to improve the data collection by the Cancer Registry, we are collaborating with Pancreas Initiative. This Pancreas Initiative is trying to create a European cancer registry patient hospital based on pancreatic cancer. In this, and we are collaborating, GRC and the ENCR, we are collaborating with them, first drafting the data collection protocol. And they are including in this protocol very detailed information about the treatment and diagnosis. For example, for chemotherapy, they are including the date, the different course, the different line of chemotherapy, the drugs, when start, when end, and so on. And for example, focus in chemotherapy, we try to use the ATC classification for coding the drugs, but we found some limitation. As I mentioned before, usually the generic name of the drug is not usually available in the clinical record. Then the staff for the cancer from the cancer registry, when they go to collect this information, it's difficult for them to identify this information and to code properly. Also, the trade name could be different among European countries. For this reason, we need to have a multilingual classification or recommendation. For example, it's difficult for the cancer registry to see how to code 5FU, because it's a commercial name. You have to go to find the generic name, and sometimes it's difficult for them to know which code they have to assign to these drugs. Also, for us, at least we don't know how we have limitation. We think it's not a standard for coding drugs combination. I don't know how is the ATC classification, but I don't know if the new drugs used in the clinical trial, I don't know if are included in this classification or not. At the moment, we don't have experience on this. But there are some limitations that is made difficult to collect the information and to standardize the data collection and coding treatment by the population-based cancer registry. Thank you for your attention.
Thank you very much, Carmen. I think that's a really interesting talk and shows about the ability to establish data collection over multiple member states, which is something that it was one of the key challenges. And we have a few minutes for questions. Are there any questions from the floor? If not, well, I'll just kick off actually to ask you, one of the points you raised during your talk, or you touched on, was data quality. Within this sort of very challenging environment, how do you ensure that the data you're getting back from each of the member states is comprehensive?
In general, for the data, cancer data quality from the population-based cancer registry, they are very well established quality indicator. And with this software that we develop, we are checking the internal consistency among the different population-based cancer registry, and also we are estimating the other indicator for completeness, validity, and so on.
Okay, so you're auditing it on a
Yes.
on an annual basis,
Yes.
or?
Yes, it's very important for us to have very good quality of the data to show the indicators.
Okay, thank you. Are there any further questions? Just one over there.
Just one information, if possible. Do you have data on biomarkers?
No.
No?
At the moment, no.
Thank you.
It's a comment rather than a question. ATC coding in the area of congenital anomalies, we found it was the absolute key to be able to do studies across different European countries. In the project Euromedicat, we actually did data linkage, linking information about a database, a European database, Eurocat, of women who give birth to babies with malformations, to actual prescription databases. And the prescription databases have all the data, the dosage, the trade name, the generic name, and we moved forward a great deal with these studies. So I know you're at JRC. Maybe when I come up to JRC, we can talk about that a little more.
My experience came from Eurocat, because before I was working in Eurocat in the Valencia registry, and then we are collecting yes, I know, but it's not so easy for the cancer registry, because the medicine the patient are taking is a lot, and we have to decide very careful which one we like to collect. Thank you.
Okay, in the interest of time, I'm afraid we must move on. Thank you very much, Carmen. Okay, the next presentation is from Nicola Ruperto, who is representing the Pharmachild project from Genoa in Italy, and is also involved with the Pediatric Rheumatology International Trials Network, PRINTO. And he's going to give us a perspective from these two projects and the registry. Thank you very much, Nicola.
What I'm going to do now is to present a very brief outline of PRINTO, just to introduce who we are, and then I will try to answer the question that has been sent in the invitation, that is to speak about the challenges that we face and some possible solutions that we have tried to implement in order to move ahead with the project. So the pediatric rheumatic diseases are rare conditions, and we can say in a simple way that they are the most common chronic diseases in childhood. And they are also highly debilitating, with effects on quality of life, sometimes for the entire life. And these are the three major pediatric rheumatic diseases I'm going to speak today mainly about juvenile idiopathic arthritis. So we founded this network, PRINTO, exactly 20 years ago with the idea to facilitate research in the field of pediatric rheumatology. And just for your curiosity, here on the right, you see a recent website that we have recently developed, which contain in more than 50 different languages information about the pediatric rheumatic diseases in a language valid for families, the list of pediatric rheumatology centers around the world, and the list of the Family Health Association. So this is the philosophy behind the research project conducted by PRINTO, which I don't have obviously the time to refer to, but let's say that for the purposes of the following discussion, it could be important to highlight that one of the first steps has been to standardize the criteria to evaluate response to therapy in the major pediatric rheumatic diseases, and the one for JIA has been in a way endorsed by the American College of Rheumatology, but more important by the Food and Drug Administration and the European Medicine Agency for all trials seeking registration in this disease. So, just as a summary slides, this table, this complex table, show that in these 20 years of activity, we have been able to collect in the different projects over 35,000 patients coming from all over the world. But as you know, well, maybe you are not all pediatricians, and so maybe you don't know this point, but at the beginning of this century, there has been really a revolution in the pediatric field by this legislation set up first in the US and then in the European Union that in a way mandate a pharmaceutical company to study drug in children whenever they want to market the same drug also in adults. This coupled in our field with the introduction of biological therapies and the existence of two large networks, one is PRINTO and the other is based in North America, it's called Pediatric Rheumatology Collaborative Study Group, has really revolutionized the field. So here you could see the patients that has been entered in through the two networks in registrative trials, so 3,000 patients. The drugs that are listed in green are the one who got a label indication, and I dare to say that without legislation, probably none of these drugs would have ever labeled for use in children. And as you could see, while the first one has been just done in North America because there was a desperate need for new drugs, for the others, the international collaboration was absolutely essential. But more important for today's presentation is maybe this point, is that we have been successful so far in convincing companies to give to these two networks the primary data outcome evaluation, which is done independently by the two networks, and then regularly transfer every month or every 15 days, depending on the company requirements, to the company for their regulatory committee with a database that, of course, has been fully audited by pharmaceutical companies. So I will not annoy you with the issue of classification, but just to set the stage, these two forms of arthritis, the systemic and the oligoarthritis persistent, we can say that are essentially typical of children, and except the first one that is very rarely seen also in adults, are only seen in children, while the other are the equivalent pediatric form of adult arthritis. So the major work in the phase 3 clinical trial, therefore, has been done in all the different subtypes of juvenile arthritis. But what is the clinical question that we have to discuss here, that is pharmacovigilance, probably started with what the FDA did some years ago, that is a black box warning for a possible increased risk of lymphoma with the use of these biologic agents called anti-TNF agents. The black box warning that then was let's say, changed over time, was based on 48 pediatric malignancies, 20 in JIA and 28 in inflammatory bowel disease, which occur in patients who have been treated with several different drugs, and therefore making, as you could immediately imagine, difficult the establishment of the related association. So some years ago, therefore, we reasoned that the effect of this drug on cancer risk is still controversial because of several confounding factors, and questions still remain, for example, if it is the disease itself or the drugs which has an effect of cancer risk, and therefore, obviously, the way to solve is a rigorous pharmacovigilance project. So if we have to make some preliminary methodological consideration, the safety from phase 3 clinical trial is acceptable, but as we all agree, is limited in terms of data. And now there are several national or company drug-specific registries, which, however, have the counter problem that has a sparse data collection of several initiatives, always or often not linked to each other. So thanks to a support from the European Union, the PI, Dr. Wolfram from the Netherlands, in collaboration with PRINTO, won a grant from the European Union to set up a worldwide pharmacovigilance registry that we call the Pharmachild. So besides the obvious question that the registry tried to answer, I think it is important for today's discussion look at this point that has been raised also by previous speaker, that Pharmachild got the ENCEPT ceiling. So the ENCEPT ceiling is given, this is the European Network of Clinical Pharmacology Unit, to registry which show independence from pharmaceutical company. In order to simplify, this can be translated in the fact that the data coming to Pharmachild belong to the network and therefore to the clinician. This has important implication, as you will see. From a methodological point of view, since this was a 3-year grant, we concentrate our effort on a retrospective data collection by revising the safety information from clinical chart, while the prospective part, which is by definition longer because we are dealing with chronic disease, is still ongoing. But this was a 3-year grant for a disease that might last for the entire life. So for control groups, we concentrate, this is maybe out of the scope of this question, but let's say that we deal with disease-modifying anti-rheumatic drugs and compare the safety events with that of these new biologic agents and with a group of patients who are normally not treated with these important drugs. So, but how we try to manage the implementation of this registry? Well, first of all, we decided not to collect the so-called mild events. For example, injection site reaction, which are quite common, are not part of the registry, but rather to concentrate on moderate, severe, and serious adverse events, and more important, what we call the events of special interest. So we try to implement strategies for the success of the registry that, as you have seen, I did not highlight, has collected so far in less than 1 year of recruitment, 8,000 patients from all over the world. We started with a census, meaning we want to know with very simple information how many patients are treated with these drugs. We concentrate on moderate to severe adverse events that physicians immediately enter in with the MEDRA code dictionary that all the regulators know very well, as well as the pharmaceutical company, and the events of special interest that I was mentioned. We try to do a simplified and user-friendly web database by providing a patient chronicle, and I will show you what I mean. But an important part has been the family involvement in such a way that we could have both the point of view of the clinician and of the families through the patient-reported outcome. But why a physician should or an health professional participate to a registry? What we have tried to do is to provide an immediate feedback by the system to the clinician in such a way that potentially they could use it in routine clinical practice with a great emphasis on patient-reported outcome and this quantitative chronicle that has been given directly to either the clinician and the families. So in a way, if you want, we could simplify by saying that we have tried to provide a service to the pediatric rheumatology community, and a service that I have to highlight is completely free. So here you can't see very well, but since we have the full drug exposure, you could see that this is the story of the drug history of a real patient that shows since the onset of the disease until the last time exactly all the drugs, the dose, and the time this particular patient was taking that particular drug. Here, there is a quantification of the level of disease activity in such a way that by eyeballing, the clinician could immediately see in few seconds the complete history of a child, and also something that we are very proud of every participating center could download immediately its own data with a database that is, we dare to say, fully FDA compliant, and has the possibility to download all the data, obviously by requesting the access to the full data to the network. The same is true for the families that by patient-reported outcome available in more than 50 languages could give their say, and now we are working on an application that will, in a way, quantify over time the level of disease activity also for them. So if we wanted to make some methodological summary, so we concentrate on the safety events that we consider are important, we concentrate on patient-reported outcome, we try to provide advantages for physician and family. The web database characteristic is listed over here, and I don't have the time to go through all the details, but as a full audit trail, a disaster recovery plan, a data anonymization for companies, and encryption for clinicians. The FDA requirements is in place. There is a full data dictionary. And as we have demonstrated for the phase 3 clinical trial, we are now in the position to directly transfer, and I want to underline, from academia to companies, I will return on this point, our data. I will not go to the results, but look just at this graph which showed the enrollment in the registry. And this second line highlights the time it takes to us to activate 114 centers. 2 years of ethics committee approval. That is the same times that we got for this trial in a very rare disease like juvenile dermatomyositis. You see, 2 years to activate 100 centers and 1 year and some months to enroll 1 child per center for this extremely rare disease. This led us to reason some years ago about what I call the so-called ethic bureaucracy, which arrived to have 104 different ethics committees which approved the protocol with only 3 refusals on the basis that we could not have been able to do this trial because the drugs were not approved for use in children. And so I hope really that this point will be solved by the new revision of the ethics committee approval. So, just for your curiosity, the preliminary results show that there is an absolutely different access to expensive drugs all over the world, and more infections are observed when we use, let's say, more potent drugs. But now let's go to the final part of my presentation that speak about the technical challenges and the possible concrete solution. So, the challenges that we face are, first of all, related to funding. So the public support, as usual for funding from the European Union, provided the initial capital supply. So far, we have been lucky to have a private support from a pharmaceutical company, but relying very much on this NSEP ceiling. So the agreement with the company was that the data is property of the academia, and they get the data in an anonymized form for the regulatory commitment towards FDA and the European Medicine Agency. So this is the registry, but here the data point that I was making, data property of academia. The Pharmachild platform is the one accepted by this particular company, and the funding has been given directly to center for data collection and also to the PRINTO coordinating center, which obviously reinvest for the overall management of the registry once the grant was over. If we wanted to symbolize what has been already touched in the prior presentation, there is a sort of triangle here that goes from academia to pharmaceutical companies and to regulatory authorities. If the link between companies and regulatory authorities is a given, we have tried from our part also to establish a link over here by having data which could be directly accessible and acceptable by pharmaceutical companies. But here, I think we have still what I can call a broken triangle, because the relationship between these two, and I'm done, is not there. And the way I think we could move ahead with this is what I could call the Food and Drug Administration moral suasion, which invite companies to collaborate with the registry, while the same, unfortunately, is not yet still true in the European Medical Agency, where personally I think a better link between the people dealing with the pediatric investigation plan and the people linked with pharmacovigilance could lead the path for the following establishment of a better use of the registry data. Thank you very much.
Thank you very much. Is there any questions from the floor? Yes, quite a lot. Right at the back, and then I'll come to the one on the left.
Hello, Christina Cabrita from the European Consumer Association. You mentioned that you transfer the data from the registry to the marketing authorization holder. How do you guarantee privacy data? And are people aware that they are sharing data? So do they give consent?
Absolutely. So here we are to distinguish two different situations. For phase 3 clinical trials, obviously the data are collected as usual in clinical trials. What we have agreed with companies, and that is clearly written in the consent, is that the data collected by the individual centers comes to the two coordinating centers, which do independent primary outcome assessment, and then the data with the anonymized criteria established at the very beginning are transferred from our database directly to the companies. For the purposes of registry, the situation is a little bit different. First of all, obviously, we indicate in the consent form that people has to sign that this data could be used for these purposes. We have a free level of a solution for anonymization. So for the individual centers could enter, if they wish, also the full data, meaning first name and last name, but these data are encrypted, meaning that in the central database we cannot understand what is, I simplify, the first name and last name of the patient. But when data are transferred, either through report or through raw data to pharmaceutical company, there is a second level of anonymization by which even myself, I cannot be able to recognize that that particular patient is coming from my own center because a new anonymized code is given to the patient before transferring.
Thank you. I think if we can have a quick question and a quick answer. Thank you.
Dirk Menzer, head of pharmacovigilance unit at Paul Ehrlich Institute in Germany and currently chairing the pediatric committee. Thank you, Nico, for presenting and your excellent work. And you know that with regard to your broken arrow there on the right-hand side, of course, we try to collaborate with you, being also regulators. But I think the biggest question I would like to have, and you had it in your presentation, how many patients are treated? And I think that is vital if you want sort of acceptance from regulators, that of course that evaluates your data collection to a useful, I would say, or utilization of the data when it comes to benefit risk or risk assessment, when we know how many patients are actually treated of the population you're surveying. And I think or even how many patients are there of the population you're surveying. I think that is vital information for regulators to utilize your data. And I don't know how much you can give input to that, but this of course leads in the end to very close link between registries and observational trials.
Okay, thank you very much. And thank you very much, Nico, for a really superb presentation. It was very impressive. Thank you. Okay, we are now moving on actually to a case study which will illustrate a collaboration between a pharmaceutical company and between an academic registry. And I'm very pleased to I should just say we're going to take two presentations in sequence. So no questions in between the two presentations, but I can take some questions at the end. And I'm delighted to welcome Jamie Geier, the Senior Director of Epidemiology at Pfizer USA, to kick off. Thank you very much, Jamie.
Thank you for the opportunity to be here. So I am an epidemiologist based at Pfizer in New York. And what I'm often tasked with as part of my department is working on risk characterization and risk contextualization. And with that, coming with that epidemiologic lens, I have some major concerns related to the choice of an appropriate comparator group. And that's what I'd like to drill down a little bit today in some of the casework that we've done. And what I'll share with you are two very high-level case examples from a methodologic perspective of what we were able to achieve in partnering with different registers over the past couple of years. And I come with a Pollyanna type of attitude that I get really excited by this, and I think there's a very real positive place for collaboration between industry as well as academic sponsors, and of course with input from regulators here. So I'm usually brought in, or the people in my group in epidemiology are brought into the clinical trial programs around phase 2, phase 2B. And the questions we receive from our clinical and safety colleagues are obvious. We're seeing these adverse events. Are these what one would expect given the indicated population? Are they higher than? Are they lower than? Are they on par with what one would expect? And sometimes we do have an internal comparator group. So that's base population that gave rise to our population that is either a placebo group or an active comparator within our studies. But more often or not, and what I've been working on most recently, is that data, that internal comparator group, that concurrent comparator group, isn't available. And I think from a placebo perspective, that's quite clear. Ethically, these data are often truncated to ensure that patients are receiving the care that is needed. So from my toolkit, or an epidemiologist toolkit, typically we go to the literature. And that is looking at observational studies first. And I think everyone in this room knows the strengths and limitations of the data. And here I'm only showing you some of the limitations. But the first is when trying to characterize what we're seeing in the clinical program, the patient characteristics of data from observational sources are not the same patients. You don't have the same inclusion and exclusion criteria that are forced upon a patient in the clinical practice setting before they initiate therapy. Additionally is the idea of time capture. And this is both calendar as well as follow-up. And by calendar, are we citing data that were published 10 years before the clinical trial program was initiated, so that treatment paradigms have changed so dramatically that we're in fact not comparing apples to apples. And then with respect to follow-up, my love of observational data as an epidemiologist is in part due to this follow-up. Long-term data understand the latency of certain events. But of course, with the clinical trial program, am I comparing again a program of 6 months' time to a study that was run over 5 years, 15 years' time? So one way we've tried to address some of those limitations is to use more sophisticated techniques, and one way is using meta-analyses of clinical trials. Again, we then have a population that's more similar to what we've studied in our trials, but you have to consider endpoint definitions and reporting conventions. In epidemiology, some of the hardest endpoints, quote unquote hardest endpoints for us, are malignancies and death. I feel very confident in that. But were malignancies identified the same way? Were they adjudicated? Were they coded similarly? And we've heard some of the limitations here already. What are the reporting conventions? And again, in clinical trials, we're faced with small numbers of patients. And if studying rare events, very low events and event rates. So it might not be possible from a meta-analytic perspective to do more there. And then there's the duration of follow-up, which I've already commented on. So one way at Pfizer we've tried to address this is partnering with registries outside of the company to create what we call in-house as standing cohorts or virtual cohorts embedded within these registries. And the advantages of this are listed here. So depending on the registry, there's a sample size that allows us to stratify on certain patient characteristics, which I'll detail in the following slides. Operationalization of endpoints. If a register, for example, is using ICD-9 codes, we can then apply those codes to our database programmatically to ensure that the endpoints are as similar as possible. And then from a difficulty perspective, there are methodologic differences that one has to consider. I was involved with a project that covered many registers and understanding simple follow-up within the registers. Do you do every 3 months? Do you do every 6 months? Do you do every year? And that's something that you have to process and own up front. But what I hope to demonstrate and show is that not only are we learning a lot through this, but it's cyclical nature, so that we as epidemiologists in industry need to better characterize our target populations. We need to understand the comorbidities, the complications, and the safety concerns. And by working with registers and embedding these studies within these existing robust databases, we are able to get data that inform our programs. And it goes on and on through ad hoc queries and additional work. So let me take you quickly through, again, it's a very high-level methodologic piece, primary care therapy A. But this was a study that was done in a primary care drug with one registry. And what I'm demonstrating here is, if you go from top to bottom, a classic retrospective cohort study in epidemiology. So we have our clinical trial program participants who are considered the exposed, patients from the register who were considered the unexposed, and looking through time at the endpoints of interest. What was very helpful in this case is that we were able to look at an overall comparator cohort of the indicated population. So to give a sense of what rates would look like in the clinical practice setting. And then able to stratify, in this case, the big variable of interest was age. So to then stratify that cohort on age and understand how similar or different these patients were with characteristics of interest to our exposed or trial criteria cohort, and then look at the event rates of interest. The second example I'd like to share was in specialty care. Again, retrospective cohort study embedded within a register setting, comparing what we had in our clinical program. This study had much more of a global reach, so we were working with several registers, which presented a lot of, I guess, strengths, but also some difficulties. But one of the fine piece of this that I found, I had a pleasure working with, was not only were we able to look at an overall comparator cohort of our indicated population, but we were able to stratify on numerous variables given the length of time the registers had been in place and the robust nature of their data. So we weren't just stratifying on age. We were studying on disease severity, gender, age, past diseases, past medications, and the like. Many of the questions, this again is in the pre-approval phase, that we had received are medications that patients have received in the past, does it make them more vulnerable for the events of interest? So not only were we able to have these two cohorts, this general population type cohort and this trial criteria subcohort, we were then able to stratify again on what past therapies the patients had. And then look at the general characteristics of the patients, see how comparable they were to our population, and then look at the events of interest. So there are a lot of learnings from these projects, as you can imagine. And what I want to share with you is what worked well. So looking back, through that lens, I think having a common protocol in place worked really well. The registers came to us with theirs, we came to them with our template, and we merged that. So expectations were set, and I also think we all parties acknowledge the strengths and limitations of the data from the beginning. Having a common statistical analysis plan was most critical. As an epidemiologist, I have one definition for an incidence rate. I didn't realize how many were out there. So getting to the table and concretizing that was amazing. And we also provided formatted tables in collaboration with the registers, so everybody knew what they had to deliver and when and how we were going to use those data. And then the common endpoint operationalization, which I touched on earlier, and we're all here face-to-face now for a reason, so you understand the importance of that. Some of the knowledge gained, and I can't stress this enough, is the adequate operational and scientific support. I underestimated how much was going to be needed in a couple of the projects that I worked on. One register, for example, unfortunately lost a lot of their administrative connections with their hospitals. In 1 month's time, went from a registry of 1,000 plus patients to one of 100 patients. So that project was halted clearly. Internally, from a programmatic perspective, having the right statistical and programming support to ensure that we can update our endpoint operationalization, that we are doing the exact same analyses as those external to us. And then the ad hoc queries happen. And I think working in a clinical trialist world as an observational researcher and trying to convince people of the benefits of these data, once they saw the data and the capabilities of the registers, the questions exploded. So I, as the epidemiologist for a couple of these projects, should have known better and built in time for those queries. And some of the challenges I think I'd like to discuss today are pooling of data for rare events across registries. We're hearing about rare diseases and rare endpoints, the capabilities, if there are any, methodologic differences across the data sources, data confidentiality, not only from the registers to the industry to registers, but I'd also, being in the House of EMA today, like to know more from your perspective about new policies, particularly Policy 70, and how, if we're working in a pre-approval space, how to protect not external data that are shared with us. And then from a timeline perspective, understand how much time is needed for these projects, not just 1 month, 2 months, but sometimes it takes 2 years from start to finish on a lot of these projects. So in conclusion, I end where I started, with this really excited idea of working with registries and bringing epidemiology to the forefront in a lot of the work that we do in industry. So I believe personally that this is a viable option for risk characterization. I think the collaborations are possible, but it's contingent on timelines. Do we need the data in 2 weeks, 2 months, 2 years? The protocols and the statistical analysis plans, recognizing and documenting the methodologic differences between the data sources, sample size, and some of the great difficulties we have with rare disease and rare outcomes, and then that support. And with that, I thank you for the opportunity to be here and look forward to questions.
Thank you very much, Jamie. As I said, I think it's best if we move on to the other perspective from Kimme Hyrich, and then we'll take questions. So Kimme is a principal investigator for the British Society for Rheumatology and Biologics Registry, and also a professor of epidemiology at the University of Manchester in the UK. Thank you very much, Kimme.
Oh, sorry. So there's been a long history now of registries, primarily treatment registries within rheumatoid arthritis, the first one launching now almost 17 years ago. And they were started at a pivotal point for our specialty with the introduction of targeted therapies, and primarily anti-TNF therapies for rheumatoid arthritis, which was a monumental step change in the way that we were treating not a rare disease, but a common disease affecting 1% of the population and a lifelong disease. And within the UK, and as you can see here now almost across 18 countries in Europe, a number of national registers of this common disease have been established. The setup of these different registers are quite different in different countries. The funding streams are quite different, but they all have at the root and the heart of them to study the effectiveness of these new advanced therapies in rheumatoid arthritis, and primarily also the safety. So we were posed with a question from a pharmaceutical company: could RA registries be used to provide contextualization data? And I think this was the first proposal that we had to use our registry data in this way. But it was a good group probably to approach, because we had already established under EULAR, which is the European Rheumatology Society, a registers and observational drug studies group. So we, independent of any input as a group of academics, had already met under a task force, and we had already a long experience of collaborative research across RA registries. So we already knew what the differences were between our patient populations. There were differences not just in the methodology, the way we captured our outcomes, whether we were linked to other national data sets such as cancer and death registers, but we also knew that there was marked differences in the background comorbidity, for example, smoking rates ranging from 9% in one country to 35% of the population in another. We know there were vast cultural differences in the approach to the medicines and when a physician would even consider prescribing an anti-TNF therapy. And we also knew there were very marked financial restrictions across different countries which influenced treatment guidelines. So the point of care for which patients would receive advanced therapies was differing across Europe. Because of this, we had agreed already an approach not to pool data, but to run serial meta-analyses. And in fact, we'd had existing success and had published looking at malignant melanoma within this group. The key issues, though, we had to face when addressing this particular issue and being involved in this particular project, I've picked out 4 key ones. The first 2 relate to agreeing a protocol that would both fit the needs of the pharmaceutical company, but that could also be operationalized within our studies. I'll talk you a little bit through the permissions and contracting, and then finally the timeline and staffing. So just to briefly tell you about rheumatoid arthritis, this is a lifelong chronic disease. In most cases, patients are treated with conventional therapy. If that fails, they're put onto a first biologic, and if that fails, a second, etc. And at each point, there's probably a drop-off of between 20 to 40% that need to move on. The whole time a patient with rheumatoid arthritis has ongoing chronic inflammation in their joints, their quality of life will decrease, their disability will increase, and their toxicity will increase. So we would expect that the adverse event rates that we might see with the first biologic, we would expect them to be higher with the second, and expect them to be even higher with the third. Therefore, we decided that we could not put all of our patients together and instead identified 3 very distinct cohorts that we would analyze. However, when the pharmaceutical company first came to us, they also asked, did you have a treatment-naive cohort? And in fact, none of the registers had that, and we agreed that this was a cohort that we would drop from the analysis. The exclusions were much more difficult to apply. Particularly, they were exclusions based on comorbidity presence. And this relates to either whether the comorbidity itself was captured in the register and whether we knew how severe it was. So for example, we would know that a patient had, say, hypertension. Hypertension itself was not an exclusion from participating. But we wouldn't know if the patient had severe uncontrolled hypertension, and therefore we had to make pragmatic decisions whether to keep patients in. We also agreed that there was a total cohort, but because in the clinical practice, patients far outside the clinical trial spectrum are included, we also agreed a sub-cohort or a trial-similar cohort. I should point out this is based on trial entry criteria, which are often very different from the actual criteria, and was not based on direct matching with individual patient data. The second challenge was to agree how we would identify the outcomes and define them and validate them. One of the registrars used data linkage to national hospital data sets and had ICD-10 codes. Other registers used MEDRA codes, and therefore we worked with the pharmaceutical company to agree a very defined list within each outcome, a basket of MEDRA codes, for example, that would be used. We also agreed that for the purpose of this project, individual event validation, so for example, if a patient had an MI, would I validate this against European classification and definitions for an MI? We agreed that for this project, this would be impossible, and the pharmaceutical company would have to rely on the degree of validation that was already going on within the individual registers. We also agreed an exposure window which was appropriate to the exposure group and the individual outcomes, and this changed for different analyses. So let me just tell you a little bit now about my own register in the UK. This is funded by the pharmaceutical industry. We currently have contracts with 8 pharmaceutical companies, and we all work in partnership to fund the British Society for Rheumatology Register. The register itself, the IP, is not held by my university. It's held by the British Society for Rheumatology, who makes decisions about how and when the data can be shared. They then subcontract to myself at Manchester University, and we're also the primary data analysts. There was obviously a lot of discussion about whether data from collected and funded by a number of pharmaceutical companies could be used to support a contextualization exercise for another pharmaceutical company. So the steps that we went through were to, first of all, agree a project with the academics, whether this was something that was feasible and could be run within the registers. The market authorization holder then made an application to the BSR Scientific Steering Committee for data access. They agreed that the data would be analyzed at the university and no individual patient data would go to the marketing authorization holder. The proposal was reviewed by the committee, and because it involved data that had been funded by other pharmaceutical companies, the proposal was also reviewed by the other 7 pharmaceutical companies that were involved. And believe it or not, they all agreed. They agreed with the caveat that no drug identifiable data and no IPD would be shared with this other company. The only thing we would share is whether it was a first-line biologic, a second-line biologic. We didn't even share the class because within some of the classes of the drugs we're using, there was currently only one product. Now, the contracting then, because the this particular MAH that made the proposal was already an existing partner with another product. We actually was very quick in contracting. It can do it as a variation to the existing contract we had. Oops. So finally, this took a long time to set up, and deadlines from the EMA, as my understanding, are not overly flexible. And it actually left very limited time to actually do the work. To give you an example, the total lead-up time from the first email to me from the company saying, is this possible, to the signing of our contract was 17 months. This left 4 months for us to undertake the analysis, which was extensive. I think we had over 200 shell tables to complete for this. And then we had a further 2 months of a final report preparation and approval. I can't stress enough how much this project relied on the existing registry infrastructure, the existing presence of data analysts already in place that knew this data inside and out, and the financial support that came for that continued to fund those people. So if I can summarize, I think this was the first time that we had worked in the pre-approval space with a pharmaceutical company, as most of our work has been in the post-approval risk management plan space. I think it has helped me as a registry lead better understand my own data, both in relation to how the data I'm being collected is being used, but also how my data differs from the data collected in Sweden or the data collected in Germany. We were somewhat limited in that we used de-identified drug data only. So although you'd know a line of therapy, it would be impossible to know how any individual drug might compare. The one thing to say is I didn't view this as an academic exercise, and I'm employed as a professor at Manchester University, and it's the expectation on me that my job is an academic job and my output will be published output. There was a considerable amount of my time that I'm not sure in retrospect was completely accounted for. And this would be something that if these became frequent requests to an academic institution would have to be balanced into how that works. And I've mentioned it already, but I can't stress the importance of the fact that this was only possible because we already had a registry up and running and had the staff in place that and available that we could then say in the next 4 months, could we move you on to this project? Thank you.
Thank you very much. So I think we've got just 2 or 3 minutes before we move on to the moderator session, just for questions around this specific set of, yes, maybe, collaborations. Can I take just 2 questions before we, yes, please.
Yeah, I had a question for Jamie regarding the collaboration with existing registries. I'm sorry, I'm Brian Robinson from Bluebird Bio. I should have introduced myself. 2 questions. One is, how did you manage the willingness of existing registries to collaborate? And 2, how did you manage dealing with differences in the quality of data that you may be getting from the different registries?
Yeah, so from a willingness perspective, to answer the first question, that was a little difficult, to be honest, because, thank you, I'm trying to find you. The speakers throw your voice a bit. So there were some people in which we had or registers in which we had had previous work or discussions and partnerships with, and that was significantly more easy. But it was difficult to get other people involved and understanding what was needed in a pre-approval space. So this is the work for the phase 2, phase 3 going in. And there were some people that pushed back, and I think understanding that we were collaborating with others, understanding the protocol, the confidentiality, and where exactly these data were going to be used helped that. So I think that transparency and contracting, it helped us there. Second question, I'm blanking, I'm sorry.
It was about the quality of the data.
Yes, so there's obvious variations according to different databases that you see and what can or cannot be collected, verified, and the like. So we really brought it down, and when we were working with several registries, to a common place of where we thought the quality of the data was comparable or as comparable as possible across. But with that being said, there are other variations in that we built that into the protocol, understanding that some could link out to cancer registers, for example, and others could not. And we just owned that and tried to present that in a discussion rather than a quantitative perspective.
Yes, over there. Have you got a mic? Just come forward and use one of the mics over there. Thank you.
Thank you. I can speak under another name as well, so I can say whatever I want. So my name is Olivier Coignon. I work here at EMA. I had a question as well on the first part of the presentation. I found it very interesting. So one of the pivotal point actually that you raised was that you had access to the endpoint basically in the registry that you used. So now if we work in a setting where you don't have the endpoints in the registries, like for instance, if you work with cancer registries, as we saw this morning, most of them, they collect data to give incidence, not even sometimes survival, so they don't have information on the treatment. So now my question is from the business point of view, if I may say, what would be your best initiative, do you think? Would it be easier for you to use the experience or existing data from cancer registries and maybe help them, fund them to collect in a prospective way information of treatment so that you will have a control arm, or basically fund your own control arm, your own registry, so that you would have a comparator to your experimental arm?
Yeah, so
Thank you.
I might be a little biased, so I think starting the discussion here and understanding what industry needs and understanding some of the strengths and limitations of the registers and just having a dialogue is where we need to be. I personally believe that experts in observational research are most, what's the word I'm looking for, are the best to run these sorts of registries, and we have much to learn from that. So if we can open up a dialogue so that someone working at BSRBR understands the queries that we're receiving, the depth of data that we need to respond in a responsible way, that would be a help. For me to go out and create another register from scratch would be very difficult, particularly in talking about things like malignancies, in which we are thinking of 5, 10, earlier today, 15 years of follow-up data that are needed. So I don't think there's a perfect solution, but there's something in between, and starting it now in a conversation between all of us is a first step, I guess.
Okay, I think in the interest of time, we must move on, but I'd like to thank both Jamie and Kimme for a really fascinating case study. I think there's lots of questions I would still have around learnings from timings, but maybe we can move that into the panel session. So I'd like to thank all the speakers again and move on to the panel session, which will be moderated by Thomas Samuelson, who's our chair of our Committee for Medicinal Products for Human Use here at the EMA, and also a senior scientific assessor at the Medical Products Agency in Sweden, and Hans Span, who's a member of our cross-committee task force on registries, and also a senior clinical assessor at the Medicines Evaluation Board in the Netherlands. Thank you very much.
Thanks, Alison. And as you mentioned, Jan and I have been asked to moderate this discussion, and that gives me an opportunity to make some remarks. The first of all, and perhaps the most important one, my thanks also to all the speakers. I think it's been a brilliant start of this session, and we're now into the discussion part of it. Maybe I should also say that being the chair of CHMP is where I'm coming from. I'm certainly not an expert on registers, but even though I've done some registry studies together with Niels Feltelius and others, but I think my perspective is very much the regulatory use and in interaction with registers, and that goes back to actually where Dr. Jürgen Kübel started. He left the room now, but he started off early on with one of the slides saying, why did we set up registers? And he talks about quality of care, and he talked about research, etc. And of course, not necessarily to ask questions from regulators. I think it's also fair to say that we regulators, through the committees headed by June Rain, Hyrich, myself, and others, we have all sorts of questions that we would like to ask. In other words, sometimes it's a matter of just ensuring quality of care, ensuring risk minimization activities, etc. But perhaps what we're challenging today and talking about today is when we start asking more questions in terms of understanding specific drugs in terms of safety and efficacy. And the real challenges, of course, will come when we start looking at products like Alzheimer's, where it will not be possible to answer the questions in randomized controlled long-term studies, where we would have to rely on registers, and I'll come back to that in a minute or two. But I think the session we've had so far has highlighted to me, and please correct me if I'm wrong, a number of key features that is needed. One of the first one was the importance of structure. Structure both in terms of data to allow pooling, in terms of endpoints, how to, several of the speakers talked about the importance of how to analyze things, statistical plans, etc. So structure in terms of data, but also structure in terms of relationship between registries holders, different registries, also between different stakeholders, rules of interactions, data privacy, ownership of data, etc. And a side remark here, I think here perhaps the regulators can play a role in facilitating and creating these rules of engagement and platforms, how to work between, in this triangle that you mentioned, regulators, industry and academia. So importance of structure, importance of sustainability, I think is extremely important, both in terms of the financial side, that was mentioned, how do we deal with that? I'm very much fond of disease registers, but there needs to be a finance here. How do we create that? But also in terms of just keeping the momentum going. And to me, the involvement of patients will be the key factor here. If patients feel that there's a value for them individually and collectively, they will push us to keep the system going. So the role of patients cannot be underestimated. The last two presentations talked about something that I find extremely important, and that's not only in terms of efficacy, I'm sure June agrees with me, also in terms of safety, and that is creating control group. Because if we don't know what to compare with, you know, the risk of going wrong is so phenomenal. And this is true both in pre and post, I take that. And all of these important structures, sustainability, control group, I'll soon finish, Alison, it's the time issue. You all talked about time, that this is not something we create overnight. And we are to blame, June and myself, perhaps being responsible, that often these questions comes up at the time of approval, limited time frame. We identify, we need to address questions post-approval, be it safety, be it efficacy, and we start talking about how do we do this? And then, you know, we're not recognizing that the registers that are out there are not fit for purpose for the questions we're asking. And that brings us to the triangle that you had. If we are to make that work, we have to start engaging early. And we, in the back, we have Professor Spiros van Varka. So Spiros, the idea is easy then to come to that we should involve these discussions early on in the scientific advice, and perhaps even before we actually have the drugs that are there. Because I'm sure June's committee, my committee, we have pretty clear ideas what we want to achieve post-approval for, or what we need to achieve post-approval of the first Alzheimer product, and we hope it all to come. We know exactly how the studies look like, and we know exactly the huge gap of knowledge that we will have at the time of approval. So we could start the discussion with the registers now, how we can set up a system 4 or 5 years before we have the, and that's the only way forward if we're going to make this work the way I see it. So I'll leave it to Jan. I'm sure you have some other interesting or better ideas than I.
No, I think that the Alzheimer is a perfect example, and I saw some people already starting to react on this. As you said, I'm also a regulator, so I look at the information at the regulatory perspective, and using a few registers already, I see that there are limitations in it, and I think collaboration will alleviate some of these problems we see. We can get from you better information, but I think that first of all, transparency is one of the key factors here, to know what every party has collected, has got on information, not to show information on different levels and to different parties and so on. So I think that is one of the things we, from a regulatory perspective, are interested in how to deal with it. How does the other parties, or the other key holders, stakeholders see it? And what I was a little bit unhappy with was the mentioning that in the various registries, the drugs were more or less taken out of the perspective, where we are interested in the comparison of the drugs and the comparison of treatment. And I see someone who is trying to intervene. May I?
Yes, indeed, I wanted to pose a question to Kimme because I was puzzled too by the fact that an academic institution accepted what I think is the biggest limitation from pharmaceutical company, not to bring forward information about the individual drugs. So in Pharmachild, once we obtain the NCEP sealing, that means we have a complete and total independence in let's say, presenting the data as we think it is appropriate for the patient, because knowing if drug A is safer than drug B, it is much more important than say that first-line drug are better than second-line drug. So I was puzzled why this, as I see, a limitation has been accepted, and if this should not be reversed with the work that we could do together, for example, with regulatory authorities.
So let me try to solve the puzzle. I think there should be a differentiation between this particular exercise that was done for a pharmaceutical company, and in parallel to that, what I do is an academic exercise. So in my academic work, absolutely, we are looking at drug-specific data. We are publishing drug-specific data. We are publishing drug comparative data. So that data is available and it's out there. For this particular company, we had to reflect back on the fact that, say, for example, unlike Pharmachild, if I understood, this is funded by the pharmaceutical companies, and you can imagine there's a trail of contracts that go behind that. And those contracts include not sharing my product's information with my competitor. And that's why for this particular exercise, we had to agree that we would look at line of therapy. To be fair, my understanding was the question is, particularly in a chronic disease like rheumatoid arthritis, we now have 13 biological therapies. Where individual new therapies might come into the line of therapy is probably more important I think it's more important to understand the patient and the comorbidity and the expected event rates at each line of therapy, as we've had a lot of experience showing that actually between the therapies there's very little difference.
So now, again, the theme is success factors, and the success factors is collaboration between registers and industry, and register industries and regulators. I'm not sure if we should try to structure it or just have a go. Let's start with some open questions or comments on all these aspects, all these relationships, and key, and share your experience, please. What you perceive to be success factors and perhaps stumbling blocks, if that's also perhaps more. Yes, please.
Hello, it's again Amanda Neville from Eurocat. I was interested this morning amongst the success factors that one of the things that was raised was the definition of a disease and then the coding of that disease. I heard people talk about ICD-9, ICD-10, MEDRA. We have a lot of different coding systems for different diseases. One of the difficulties in collecting data in a European setting on congenital anomalies is the use of these different coding systems. Eurocat collects its data in ICD-10, whereas the Italian medical system, the whole hospital system, uses ICD-9. So that means that registries in that country have to use two coding systems, which is very complex. How do we move forward on this? Well, in EuroCap, what we've done, and this is something I would suggest to other registries if you don't have it, is we have a coding and classification committee. So we look at our data and we look and see what is the definition of that disease, what is the most appropriate code, and we make recommendations to our member registries to use a particular code for a particular disease. And this is an ongoing process to improve the quality of the coding and the classification of the diseases. And of course, it's an open issue, which if we can collaborate in any way in improving coding in the ICD systems together, that would be wonderful.
Any remarks from the panel here? Some of you spoke about this.
No, maybe just a minor specification. We have to distinguish, of course, the classification of the disease by ICD-9 or 10 from the MEDRA coding, that is a completely different stuff, is related to safety events. And this is a dictionary that I think is worldwide accepted and that could be used across all the registries without, I would say, almost any further discussion. So the 2 points are quite separated.
Is this, and now I return the question to you, is this an invitation to get set up a group of shareholders and to make, let's say, rules how to set up registration from that perspective and, of course, other perspectives?
Sorry, was that a question back to me?
Yeah, it's the question.
Yes.
This is: do we feel that all people in this room, of course, is there a necessity to have a force, a group of people that can define the characteristics of a register? What kind of ICD you use? When do you use it? Do you use MEDRA for side effects? What MEDRA do you use? So, is there a necessity to have such a group?
Can I ask a similar question? I would imagine that there are huge differences depending on which therapeutic area you're in. And in some areas, there are quite good harmonization on what to use, etc. And others, there's not. And I guess in some areas, with the Learn Society, European-wide Learn Societies that works for harmonization. So I guess it'd be very difficult to do something general along the lines that you asking, John. What I do think, and here it's again a question to the audience and the panel, the role of regulators when it comes to what I call rules of engagements or find ways of describing relationship between register holders, MA, industry, regulators in terms of data privacy, financing, different models of analyzing things without moving data away from registers, etc. So to create these types of structure more in detail, whether that would be of value if EMA starts doing.
Thomas, maybe just a comment on that,
Yes,
because a certain amount of work has already been done.
That's why
with a kind of regulatory catalyst, not necessarily done by regulators, and that's in the context of the European Centre for European Network for Pharmacoepidemiology and Pharmacovigilance, NCEP. And in fact, Nicola referred to this in his presentation. So there's a code of conduct which sets out, if you like, best practice for how funders of research would operate and have relationships with those conducting the research, and also sets out standards in terms of transparency, publishing data management, if you like. There's also ongoing IMI projects that are working in this space, I think particularly of the Advanced project, which is looking at benefit risk studies in vaccines, but again, looking at these fundamental principles of governance, be they transparency, independence from funders, etc., managing conflicts. Whether we need something specifically, so a number of the presentations in the first session said perhaps we don't need more rules, but perhaps the implication was, could we distill the core and apply them to registries? And I think that would be a question that could be very interesting to see if people thought that some kind of distillation on core governance principles, as well as data principles, might be of use for fostering collaboration between the different stakeholders.
Peter.
Yeah, sorry to speak as a regulator, but I was picking up and wanted to bring it.
Why do you apologize speaking as a regulator, Peter?
Well,
you put us all in the trouble.
No,
here.
I want to. This is my problem with English. I want to say sorry to speak as a regulator again, but I think what I'd like to pick this up is indeed no more regulations. That's what I heard in the first session, and I was thinking like, okay. What could we do to move ahead? Because I think having one universal database which has all the data connected in one singular way, that's a utopia which we will not reach. At least I'm not convinced we will do that. But what I think is very interesting, and I picked it up from Dr. Jamie Geier's presentation, is that we should start planning early on, because that's what you also said, Thomas, but you mentioned it. We start around phase 2 to think, okay, what are the problems? And I think when you started talking with the different registries, you found out that you needed to map what is the different ways of how they record the data. Is that not something that we could sort of agree on as a sort of common idea? If we develop a new drug, we need to understand the disease area, what are the important characteristics or the important codes or outcomes that we need to collect, and what's around in ICD-9, ICD-10, MEDRA, whatever. And I think that's a way we could more structurally address and move on. Then we don't solve the whole world at once, but at least around my one product, we know we come in, and this is the way how we could proceed, looking at observational data.
As you heard this morning, I'm a supporter of the distillation principle. So no more rules, no more groups. No, but I think one method to go forward is to lift up the successful examples and look how it has been done. I think they are out there, and I think they have sort of challenged all the problems we have discussed here, and some have solved at least some of them, and another registry have solved another. Look into that.
I would like to expand on your comments and on Peter's comments by sharing with you two sort of semi-confidential information without obviously referring the source, but it happened to me that speaking with two companies in the very early phase of development of a drug, I mean pre-pediatric investigation plan submission, I was told by one company that when I asked, what about pharmacovigilance post-marketing authorization, the response was, I will deal with that once I will get the marketing authorization. But the second one was much more interesting, and this is I want to share for the comments you make. Let me say, currently at the European Medicine Agency, the two offices, pharmacovigilance and CHMP, PDCO, don't speak to each other, so why I should make this link between them? So I make this as a provocative proposal to state that your comment about the early involvement, I think, is really the key. So if there is a way, absolutely with no new rules, because we have the rules that we need, by which what is going to happen after the marketing authorization and for pediatrics after the completion of the phase 3 trial in the very early phase of the discussion, I think this will serve quite a lot the work of registries.
I think that's good, and Peter, you apologize, you poor English, and I should perhaps, when I meant rules, I didn't mean rules in regulatory terms, I more frameworks, and I was also asking whether there's a need to develop methodology here. So this is not, this is an open question, but there are the methodologies in terms of statistical analysis that would decrease the data privacy and ownership discussion issues when pooling data between different registers. And I think also here, these types of statistical methodological aspects is something that could be something we could further explore jointly, academic and regulators together. Now, thank you, there's a gentleman in the back, and there's two hands in the air, but let's start with you, gentleman.
Thank you. I'm Giulio Maria Corbelli from the European AIDS Treatment Group. Following this suggestion of improving the collaboration among stakeholders and also your recommendation about not underestimating the contribution of patients, I was asking myself, how can we motivate patients to participate in the inpatient registries? The example of PRINTO was kind of interesting to me because I appreciate that the physician, the medical staff has the advantage to get some feedback immediately from getting the data into the registry. But I was asking myself, how could you provide some kind of feedback to the patient? By the way, I could not see in the slide how was the participation of patients in PRINTO? Was it satisfactory, and could you get any feedback about how happy they were about participating?
So for Pharmachild, because of the time limitation of the grant, we concentrate mainly on the retrospective charts collection that was done through the physician. But for the prospective part, that start with the private funding, once the grant was over, the participation of the family was the key issue. And I have to say that so far we did not experience any problem at all, because the system we have developed is done in such a way that families, and for families I mean the parents in our case, or the child himself or herself, could complete or should complete and do complete their patient-reported outcome even few days before coming to the clinic, or while they are in the clinic waiting for the physician to performing the visit. So the system that is managed by a nurse, which sends an invitation just a few days before the child comes to the clinic, in order for them to complete the patient-reported outcome at that time, or if they don't do, again, as soon as they are registered in the clinic, they complete the patient-reported outcome while waiting for the clinic visit. In this way, the system, I have to say, has been extremely successful. Now we are moving to the second step, that is to provide a feedback to the family himself by showing in a simplified format how the child is doing over time for the level of disease activity, in such a way they could have on their tablet or smartphone or whatever you want, an overall summary of the situation of the child.
Yeah, I see your hand, but I think this is exactly I'm happy that you're saying this, because it more or less confirms what I naively thought, that involvement of patients and the patient's feedback to patients is essential in the future. And you also say, let's look beyond the paper solution. Let's see what the new technology can allow us to do, and use of apps, phones, whatever, to report these things, but also to get feedback on an individual level. And again, it goes back to Jürgen. I referred to you earlier when you were out, but you displayed why you set up registers to ensure good clinical practice, to ensure to meet research questions. But more and more, of course, we have to set them up or address questions, the need for patients, because again, I think that's important for the sustainability. I'll soon go to you, but I had a hand in the back of a lady. Yes, please.
Cristina Cabrita from European Consumer Association. I have more questions than solutions. And one of the questions is the use of registries. What do you think about is the use of registry for every disease? Which type of disease, which type of medicine? Is there a guidance? That's one of the questions. And the other one is the registry purpose fit, like it was told before in the morning. Because if we had a lot of purpose, we have to gather a lot of data. And as Jürgen said, a lot of data doesn't mean quality data. So that's there are my questions.
Well, but because you're at the EMA, I think we need to start asking that question from a more regulatory perspective. Why register solution? Why do we need registers, and when do we need registers? And June, is it fair to say, I tried to outline that before, that it all depends on the given application, the given situation. In some situations, we've had situations where we say we want all patients treated with this new drug to be included in a register. Because that ensures quality, that ensures compliance with risk minimization activities, we can follow it, etc., etc. In other situations, we say we don't need registers for this. We have other tools to answer the questions that we think. So I don't think one can answer the question I can't answer the question in a general way. Yes, please. But of course, from an academic or clinical perspective, it's totally different.
I just want to address both the questions at the back and also Nicola's. I think the utopia of a register for every product and every disease, and each individual register being the utmost quality with the best quality safety data and high-tech feedback to patients doesn't come for free. These are exceptionally, for the highest quality registers, these are registers that aren't £10,000 a year to run. These are running into the millions of pounds, and that money's not available. Within the UK, we get feedback from our charities, from the MRC, from the NIHR, that they're not there to fund registers just for the sake of having a register. So that I think that has to somehow fit into the equation, that what the ideal is for how we can monitor drugs in real world or others has to be balanced about what we're actually able to do.
Absolutely, and I think I mentioned in financial aspects in it. So it needs to be based on the real need to address a question. Absolutely, Jürgen, and then Francois.
Yeah, just as a comment, because you said you have already now in your mind what you would like to see in 5 years, and you would like to start with phase 2 already, talking also to registries. To be honest, I think this is already too late. Because if you think about the process of implementing this into the registry and then capturing real-life data, at the moment you would like to need them, we might not be ready. So really having sort of in mind what for certain diseases you have in mind you would like to generally see with whatever drug which will be developed would be more helpful than approaching us at phase 2 of a certain defined drug. Rather tackle different diseases and say, for this disease, we would like to see this minimal data set.
That's well taken. And I explained the only reason why I said 5 years is the most optimistic time frame before we see a new Alzheimer product, and that's we don't have. If we're lucky, that's the time frame we have now to start working on that. We should have started earlier. That's a good point. Absolutely, and of course here it is, and to go back to an earlier comment here, it may also be just understanding the epidemiology. How does the patient population that we intend to treat, the MCI patient population, actually look like? Because the registers, at least that I'm aware of on Alzheimer, they don't capture that at all. Francois, I think you get the last comment, and then Jan as a moderator.
Yeah, François Meyer, HS. So my question to the speakers on the panel is about the nature of the data that could be collected with regard to drugs on these registries, because my impression was that we had more data related to the safety of the drugs rather than to efficacy or effectiveness. So is that a right impression? And is there any limitation in the acceptability of non-randomized data issued for non-randomized clinical trials, I mean for other sources than randomized clinical trials, to conclude on efficacy effectiveness? Or can we see registries as a good way to help to evaluate the relative effectiveness of various drugs in a therapeutic field?
I think it's vital to have some picture of the disease activity, which vis-a-vis can equate to be calculated into the treatment effectiveness, and it has to do with the confounding by indication. And remember that the drugs are given into the patient, and the environment that you're giving it into also has to be captured. Otherwise, you can't make any sense of the safety that you're capturing. So I think there's a lot of registers that are set up and do have to capture effectiveness data. So I think they can deliver on both fronts.
I don't know if we can answer it very quickly, but I wanted to touch back on timings issue that both of you raised in your presentations, and whether there were any learnings specifically from your interactions, and maybe Jonathan as well, about when can industry start to engage with an academic register, and also from the perspective of an academic register, how long do you need in order to engage? And then have you any learnings which could facilitate this process going forward? I don't know if you can quickly give us a view.
Yeah, I think, something that's worked really well where I am is engaging with registers very early on. So for me, it's the phase 2, phase 2B, and one of the examples that I showed you, by setting up these virtual cohorts of patients that were similar to ours, we were then able to, when we received a query from the agency, respond in 1 month's time. So, we had this from a background contextualization, but then in more real time, the capabilities to do so. So working from that and thinking of registers not only as a post-approval pharmacovigilance tool, but also as something that we could use early on to understand the epidemiology of the disease, not just drugs, but the disease, I think, is a first step.
I think part of the answer probably relates to what already exists for the register. So if there's a register that's already up and functioning and has taken on that role, then the phase 2 stage is probably the right time. We've even had companies approach us at the phase 3 stage, as long as we can agree by the time the drug is on the market. Otherwise, you're going to miss exposure. I think if you were looking to set up a new register in a disease area that didn't have one, 5 years plus.
Rob Hyde from Biogen. Just a couple of comments. One, I think your comment about Alzheimer's disease is a very relevant one. And the one thing that is happening, there is an IMI call in IMI2 under the Big Data for Better Outcomes called Roadmap, which involves a number of the companies and leading registry leaders to try to map what is needed. So I would say if the EMA have and the payers and HTA have investment in that will help guide what we can do for Alzheimer's in the future. Another comment is the paradox at the fundamental coalface is the physician time, and the physician time to enter data or to be engaged in recording the data. So the simpler we can make it with core data sets and minimal safety information to expand our knowledge, the better. And I think if you and we can come up with that, I think it will really help registries to be more viable and more valuable.
Thanks. I think that's very good comments. Francois, to go back to your question, my sincere belief is that absolutely registers must be able to deliver answers also in terms of efficacy and relative effectiveness. Not as an alternative to randomized controlled trials. I'm very much a wholehearted randomized controlled trial person, but just recognize that there are things that we can't address with randomized controlled trials for a number of feasibility aspects, or as you say, there may not be an incentive. We've had in Sweden a very nice proposal to do a relative comparative between the different new anticoagulants in a register, something that would never happen unless it's done by if it's done by companies or something like that. Unfortunately, that failed because it costed £8 million, which is a peanuts actually if you look at it. But okay, that's where it goes back to the money issue. But absolutely, I mean, if there are considered to be generate robust safety data, there must be a way to generate robust efficacy data. But Jan, you get the last comment now before lunch.
No, it's not a comment, but an observation. I've heard to start early and with a register. And I've seen in the lysosomal storage diseases, after a few failures, we now start with a natural history as a register. And there we learn, at least we, the company, the physicians, patients learn what is the normal at that moment, what is the normal nature of the disease, what is expected from the disease, and what are the most important endpoints. And I think that is a nice model that you can take in mind when going further. That delivers, solves a lot of problems. It solves the problems, how do we classify it, what do we use as a reference, how we have the epidemiology. So I think that's a nice model, and maybe we can use it more if the disease and the drug gives a reason for it.
So it remains only for us to hand the floor back to you, Alison.
Okay, perfect. I'd just like to thank all the speakers again and also the panel moderators. I think it was a really fantastic session, so thank you very much.